right? Because like I said, this is nothing more than a review of everything that we've been talking about with hair so far, with a couple new little things in there. So at one point, we looked at the structure of how hair grows in the first place. And there's a bunch of cells here that help to produce that. What was the name of the protein again? To, to produce keratin, which is the protein that makes up your hair. And then it keeps pushing it into the hair, and you get more and more hair made. Where's the newer parts of the hair? In the roots. The stuff that's coming out that we can see right now, that's old stuff. The newer stuff is still inside of your root of your hair. We talked about the fact that your hair has 3 layers. You have the cuticle, which is the outer covering of the hair. You're going back to the pencil analogy, it's the paint that they put onto the pencil. This is formed by overlapping scales, which point towards the tip. And you can see this particular example in the picture. Is this a healthy hair or is this a damaged hair? Pretty heavily damaged because the cuticle's flaking off pretty bad. All right, the scales are going to originate from the follicle. And remember, the cuticle is important in forensic hair analysis because it helps to identify the species from which the hair most likely came from. Then the next layer, which is like the woody layer of your pencils, that's the cortex. So this is going to be just under the cuticle. This is where all the pigments are for causing you to have the hair color that you have. And what's the name of the pigment again? Hill, you said it the last time I asked. Good. It's melanin. The name of the pigment is melanin, and I am going to hold you accountable for that. So you should be aware that the pigment that's causing your hair to have the color it has is a pigment called melanin. And then this is where most of the keratin is as well. This is the part when we do make associations. You notice I'm not saying match. I'm saying associations. If we say this hair is closely associated to this other hair, it's really the cortex we use for making that determination. And then we use the medulla as well for species identification primarily, and that's the central canal that runs through the hair. And then we saw that we can do measurements with that and compare it to the diameter of the entire hair, and we'll get into that here in just a moment. So when we talk about the cuticle, that outer layer, there's 3 patterns that we can see. We can see this coronal layer, or this coronal cuticle pattern, which I think looks like a trapezoid. We also talk about being the stacked cup thing. You have spinous, it looks like what you think of with the spines on a reptile, and then imbricate, which we saw in our own hairs, right? And these are going to be the more important things in helping you to do your quiz the next time that we meet. Because the next time we meet is the day I'm going to have the microscopes all set up for you already, and you're going to go through and try to identify what species it is that's under the microscope, or what species the hair that's under the microscope was taken from. And then there's going to be other questions to answer with it. You will be able to use your labs. You will be able to use your notes on the quiz. You may not use any other outside electronic resources. So I'm not going to let you guys connect to the internet to be able to look stuff up. So your notes or anything that is on paper in your possession, you will be able to use on the quiz. Capiche? They said yes. All right, so then you have your medullary index. This is a measurement that we can do that helps us to identify the species from which the hair may have possibly come from. All we do is we measure the width of the medulla. So you can see they got the ruler there. And we compare it to the width of the actual hair. So the width of the medulla here is about, I'm going to assume this is millimeters, is about 3 millimeters. Actually, it might even be micrometers. Almost go with micrometers. 3 micrometers compared to 10 micrometers for the hair. And so that gives you a medullary index of 0.33. Typically, human hair has a medullary index of 1/3. Other animals, other mammals are typically going to be 1/2 or in some cases larger. And then we looked at the different medullary patterns that you can have. So when we look at the medulla, and I noticed when I was looking at some of your notes from when you watched my last video, a lot of you didn't have this written down. So if you don't have this, you're going to want this because this will show up later on. There's 4 patterns in medulla. There's the continuous pattern where you have the medulla throughout the entire hair. There's the interrupted pattern, and in the interrupted pattern, you get sections where you have medulla, then you get sections where you don't, then you get sections where you do, you get sections where you don't. And there's no set pattern in those particular sections. Whereas in fragmented, you get relatively even-sized sectioning of medulla with spaces in between them. So interrupted is pretty random, whereas fragmented has its own set pattern for the medulla. And then there's absent, where it's typically transparent and you can't see that it's there. All right, most of the time when we see human hairs, it has no medulla or is fragmented. And so most animals are going to have continuous or interrupted medulla. So any questions there? I see a lot of people writing stuff down. You guys need a moment then? I'm going to pause the video a second. And we're going to pick back up from here. So then, quick little video I want you guys to check out. The forensic scientists testified that the hairs found on the victims were most likely hunters. But they be sure the hairs were hunters. Hairs simply aren't distinct enough to be linked to an individual. Dr. Kurt Forrest is one of the world's leading authorities on the forensic analysis of hair. He was called in to examine the hair from the crime scene and prepare conifers. Forrest first looked at the hair's color. Color in hair is determined by tiny pigment granules that appear here as brown speckles. Adjusting focus lets the forest examine the end of the hair shaft for pigment distribution. In some hairs, the granules are distributed evenly. In other hairs, they form clumps. Another characteristic of hair is its outer sheath, called the cuticle. The cuticle grows in overlapping scales, like the skin of a fish. These scales can be pointed, rounded, or square, and can lie very flat or curl up. Some hairs have a clearly visible inner shaft called a medulla. In other hairs, the medulla appears flat and broken, or they have no medulla. The Forrest looked for similarities between the hairs found on the victim and the hairs taken from Ed Honiver. He saw some similarities in color distribution, but he couldn't create a trace-for-trace match between the crime scene hairs and any of Honiver's hairs. All right, so good overview of how they can use this. It's pretty similar to the video that we just watched as the opener too. So any questions there? Once again, the key is we can't say we have a match. We can say we have a certain association. So because of me being a stats geek, I got some pretty, what I think are fun stats that go with this. Redheads, you have less hairs on your head than anybody else in this room, on average. And my wife's the same way. She sheds worse than our dog. And I'm not saying that mean. I know sometimes I say things on purpose to be mean to my wife, but no, I don't do that. Come on, you guys, that was a joke. Anyways, yeah, I can relate because her hairs are laying around in the sink all the time. I have to take our sink trap apart every other year or so because it gets clogged up because her hair sheds so much because she's a redhead. All right, black hair is typically a little around 100,000 hairs per head, whereas a blonde, you typically have more hair as a blonde than anybody else. For most people, the maximum length of hair that you will get is 3 feet long. Once it reaches 3 feet, it's not going to grow anymore. That's not true for everybody, but that's for most individuals. Annual growth rate of 6 inches per year. Some people grows faster than others. I know mine grows faster than that, at least where I have hair. Female hair grows more slowly than male hair. Guy hair is typically more dense. This is the one that throws me off. More dense than female hair. That one surprises me. And then I look around and I look at some of the guys in here. You know what? That makes sense. You have some pretty thick hair. And the diameter of a typical human hair is usually no more than 0.1 millimeters. That number seems a little small, but all right. Now here's where some of the new stuff is coming in. Your hair grows about 15 millimeters per month. Right, so I would like you to know that stat. About 15 millimeters per month, which is about half an inch. And during is what we call growth phase, or what you'll hear in science, we call it the anagen phase. So I want you to be able to link the anagen phase as meaning growth phase, and I want you to be able to recognize that the catagen phase represents the scientific name for when the hair is dormant. All right, so once it's gone through the anagen stage, which is usually the first 1,000 days of that particular hair, then eventually it's going to become dormant, and then eventually it's going to fall out. When it's at the stage when it's going to fall out, we call that the telogen stage. So you can see the anagen stage is typically going to last about 2 to 6 years. Then it's going to start going into catagen stage, where it's going to be dormant, and that's going to last for a couple weeks. And then it's going to go into the telogen phase, where at some point it's eventually going to break off. And now you're going to start getting, you notice, new hair starting to grow in. And so then it'll restart the anagen phase, catagen, telogen. Then after it breaks off, you'll get the anagen phase starting again. So it's a cycle that continues over and over and over until you get old and it decides to not start growing back, which stinks. Trust me, I know. All right, so growth goes with anagen, catagen goes with dormant, and telogen goes with the point when it's starting to fall out. Questions with those? There's been a lot of studies trying to figure out why, for people that are balding, why the anagen stage does not start back up at a certain point. We know it's a genetic thing, but the reasoning behind why, they're still studying a little bit. I'm sure they have. I haven't looked it up in a while. I'm sure they have a better idea now because of the Human Genome Project, but I don't know for sure at this point. All right, so then we spent time the other day talking about how we collect hair evidence. We use it for association and exclusion purposes only. We can't say it's a match. We talked about some of the ethnic generalizations, and we talked about some of the different areas of the body where the hair originates from as well. So when we collect hair evidence, remember we have to collect 24 to 50 controls for doing the analysis. 24 for what type of hairs? Any hair from the body. I heard arms, I heard legs. People are still a little afraid to say pubic hair, but that's okay, I get it. Oh, somebody said, okay, they're not as afraid. Good. Your trunk hair, your chest and back hair, those are 24. What's the 50 go with then? That's hair from your head. All right, when we collect these for control purposes, we have to get the entire length of the hair. It can't be a partial hair. It has to be the entire length. And then through this one in, as well, in sexual assaults, you got to comb off all the foreign hairs and place in separate containers. So when you take the hair samples, if there's a foreign hair that seems to be mixed in with that, with the hairs that you're collecting from your control, you want to separate those for separate analysis. And then victim sample hair should always be obtained, and I think that makes sense based upon all the conversations we've had. So when we look at these and we make these comparisons, we're looking at the color, we're looking at the length, we're going to look at the diameter, we're going to look at the medulla patterns, we're going to look at the distribution of the pigments in the cortex, and what we're really looking for is what are things that are similar between these. Other things that can help us, morphological abnormalities. That is a big, nasty term. What does it mean? What's morphology? It's what, okay, when the Transformers do morphology, what are they doing? They're changing their what? They're changing their outer appearance, aren't they? That's what morphology is, outer appearance. So if there's abnormalities in the outer appearance, isn't that going to make it easier to identify where that hair may have come from? If they have something weird with their hair, that's going to make it easier to identify if it might closely resemble hairs found at the scene of the crime. Deficiencies, infections, you can get fungal infections. What's an example of a nit infection? Anybody know an example of a nit infection? Happens in a lot of elementary classrooms. Lice is an example of a nit infection. So you can look if there's presence of lice or something similar to lice, that can help you to make some comparisons here too. This is a graphic I didn't get a chance to show you guys the other day because we ran out of time. They can open it. All right, so when we look at the persistence of hair, take a moment and just analyze the graphic. You're okay. So once again, you guys, take a moment, focus on the graphic, not Amber. Take a minute, look at the graphic, and see what you can figure out there. I'll be quiet here for a few seconds so you guys can analyze it. And we want to try to summarize the graph? Just a general summary statement of what this graphic's trying to show us. Oh, am I going to have to deca-doom it? Chris, give it a shot. What's the trend? See if you can tell us what the trend is telling us. Okay, so what does that mean? Because you're right. Oh, thinking a hat. I can see that. All right, let me help you out with what time of wear means. What that means is the time since the crime occurred. And what does it mean if it's less persistent? What do we mean by persistence? Tyler? Yeah. So the more time goes by, and we can see that here, the more time goes by since the crime actually happened, the less likely you are to find hair evidence at the scene of that crime. So if you get to the crime scene immediately, you have a much higher likelihood of finding hair. Otherwise, if you don't find that particular scene until days later, the chances of you finding any trace evidence, not just hair, this could be fibers, it could be gunshot residue, any small trace evidence, the persistence of it being present at the scene of the crime decreases significantly as time goes by. And then I like this one too. The hair analysis costs a lot. So we're talking a small case here. Just to run simple testing to get as close to 100% accuracy as possible, to run 2 samples against 2 known samples costs around $10,000 if you want to do the full analysis to get as close to 100% likelihood of those hairs being associated with one another. That's kind of expensive, isn't it? So do you think many police departments and agencies use this? No, they do the simple analysis to see if there might be association, and that's about it. Unless they can pull DNA from the root of the hair. All right, then the last thing we've been talking about is we've been talking about some of the the ethnic characteristics of hair. And we looked at with Negroid, it's typically going to have more of a coiled appearance. The pigments are densely distributed, but they're going to be scattered throughout. And the cross section is going to be a little more flattened. Whereas Mongoloid, the appearance is typically straight. The pigments are densely and evenly distributed, and the cross section is going to be round. So this picture here, by the way, is showing you the cross section. So if you look at the cross section here with Negroid, see what we mean by flattened now? Whereas when you look at Mongoloid, relatively circular. Caucasoid, look at the variations. Some are round, some are oval. All right, and then with Caucasoid hair, typically generally straight or wavy, they're going to be more evenly distributed pigments, and the pigments are typically smaller, and you're going to get a variation in the cross sections. And then we looked at some of the areas of the body as well, and I told you don't worry about the areas of the body as much as some of the others. A couple of the highlights, though. Head hair, we know it can be straight or curvy, can be wavy. Pubic hair, we said we look for the presence of buckling, where it looks like the hair is kind of snapped a little bit. Facial hair, look for double medulla. Chest hair is typically going to be pointed, similar to pubic hairs, because of all the abrasion going on with their clothing, and so on and so forth. All right, so that should hopefully tie up any loose ends that we may have with the hair stuff we've been discussing. Are there any questions that you guys have? I told you 20 minutes, and look at the counter on the recording. That's 22. I think that's pretty good.